I know the name of the channel is Psych and Boba. Finally got the boba. On a Friday evening in 1973, Candace Pert was busy working after hours on an experiment, learning among the brightest minds in clinical research. But why couldn't this work wait until after the weekend? Was she this obsessed with psychopharmacology? Well, not exactly. The answer was a bit more simple, but also a bit more intriguing. This passion project was a secret. So what is it that Pert would end up discovering? Why was this a secret to begin with? Let's dive in, shall we? The brain and nervous system are made up of billions of nerve cells, called neurons, and it's through their extensive network that messages are transmitted to, from, and within the brain. Each neuron has its own dendrites, parts of itself dedicated to receiving messages from its neuron neighbors, sent in chemical form. On these dendrites are receptors, special molecules that recognize specific chemical messages. Different types of receptors recognize different messages. For example, thermoreceptors pick up and help the neuron pass along messages around temperature. And photoreceptors do the same thing for light. But Candace Pert wasn't interested in temperature or light. Instead, she had designed an experiment to find opiate receptors in the brain. Ever since German scientist Friedrich Serturmer, sorry if I butchered that, first isolated morphine from the opium poppy plant in 1803, this substance and its derivatives saw widespread use in clinical settings as effective pain relievers. You probably know these chemicals as opioids, drugs that include names like heroin, oxycodone, Vicodin, methadone, and fentanyl. A graduate student at Johns Hopkins University, under the mentorship of psychiatry and pharmacology professor Dr. Solomon Snyder, Candace Pert herself came to appreciate the clinical use of opioids after a riding accident resulted in her being treated for a broken back, an experience that would help shape her scientific interests. However, the reason that Pert, or any of us for that matter, could even feel effects from opioids is because when these chemicals enter our bloodstream, our opiate receptors pick them up, and in turn, cause a series of neurons to pass on messages that tell our bodies to feel pleasure or pain relief. Initially, Dr. Snyder had actually discouraged Pert from doing this research. He himself was unsure of whether finding these receptors was even possible. After all, based on similar research in fish, it was possible that these receptors only took up tiny parts of the human brain. I mean, it's understandable that the head of a research lab wouldn't want to spend all that grant money on what he considered the equivalent of finding a needle in a haystack. Pert, however, had other ideas. She went ahead and ordered lab supplies behind Dr. Snyder's back anyway. Needless to say, his finding out about this could spell trouble. That Friday evening, Pert injected radioactively tagged morphine into a sample of brain tissue. She let it sit over the weekend, hoping that this morphine would eventually find its way to the tissues that contained opiate receptors. Her hunch was correct, and what she found on her lab desk that Monday would also spark a new path of research for Dr. Snyder's lab. However, Pert and the Snyder lab weren't the only researchers busy discovering receptor sites in the brain. Around this time, Dr. Lars Terenius's research group, based in Sweden's Uppsala University, and Dr. Eric J. Simon's research group, working out of New York University, had also independently found brain receptors specifically for opiates. Together, this body of findings pointed to a new question. You see, our bodies don't actually make heroin or morphine on their own, so why did the brain know what to do with them? The 1973 opiate receptor breakthroughs strongly suggested that there was a substance similar to morphine that was naturally produced in the body. And in the mid-1970s, the race was on to identify exactly what that substance was. The winners of this race? British neuroscientists Dr. Hans Kosterlitz and Dr. John Hughes. Working with pig brains out of a small, underfunded lab at the University of Aberdeen in Scotland, and using a screening test developed by Dr. Kosterlitz, the pair narrowed down the various candidate substances to 2 peptides. Dr. Kosterlitz would name these enkephalins, based on the Greek term for in the brain. Their chemical structures confirmed that, on a molecular level, they contained parts that were similar to opiates, and would allow them to attach to the opiate receptors that had been discovered just a few years earlier. However, enkephalins weren't the only substances with these features. As it turns out, by finding the first enkephalins, Kosterlitz and Hughes had discovered the first of many types of what we know today as endorphins. Endorphins are produced by the body's pituitary gland and hypothalamus when your body needs to counteract stress or pain. As you can probably guess, they act as pain relievers. After all, the name endorphin itself comes from a combination of the French word endogène, meaning produced from the inside — my French is non-existent — and the word morphine. So together, self-produced morphine. Endorphins trigger a soothing, pleasurable mood lift when they're picked up by opiate receptors like the one Pert discovered. As it turns out, opioid drugs work because their chemical similarities allow them to mimic endorphins attached to opiate receptors and trick our bodies into creating the same effect. Even after the initial enkephalin findings, scientists have gone on to discover more classes of endorphins. For example, in 1978, neurochemist Dr. Cho-Hao Lee at the University of California, San Francisco isolated a different type of endorphin, one multiple times more powerful than morphine, beta endorphin, from camel pituitary glands. Today's fun fact: the 500 camel pituitary glands that Dr. Lee used in his research, well, I guess the fact that he used 500 camel pituitary glands isn't itself a fun fact, but bear with me. The 500 camel pituitary glands he used just happened to be sitting around in his storage since the 1960s. Yep, they were originally intended for a different project on fat metabolism. Those 500 glands never quite helped Dr. Lee find beta-lipotropin, which was his original goal. But I'd say stumbling across a new class of endorphins more than makes up for the investment. Not to say fat metabolism isn't important, but I mean, think about it. Have you heard of beta-lipotropin before this video? Didn't think so. Okay, back to endorphins. Today, it's believed that there are more than 20 different types of endorphins. And beyond understanding the types and their functions, researchers have also put a lot of work into understanding how we naturally release endorphins as well. Their release has been linked to activities like exercising, massages, acupuncture — by the way, we have a now Dr. Pert to thank for discovering that — sex, intense laughter, music, and even eating dark chocolate, which probably explains why you feel good after doing many of these activities. Additional research has also been done to better understand the workings of endorphin synthetic cousins in the hopes of finding ways to balance managing severe and chronic pain with mitigating the dangerous risk they possess for addiction and misuse. While there's still much to learn regarding endorphins and opiate receptors, endorphins seem to be our body's natural way of coping with adversity, temporarily increasing our threshold for stress or pain in difficult situations, and allowing us a way to thrive in spite of it all. So while I spent this video covering how science came to discover endorphins and its pleasurable effects, I have to end this on a somewhat depressing note. The discovery of the opiate receptor would win the Albert Lasker Award, second only to the Nobel Prize, in 1978. This award was shared between Hans Kosterlitz, John Hughes, Solomon Snyder, and that's it. Apparently, because Candace Pert was just a grad student at the time, in one of the most painfully ironic twists in modern neuroscience, recognition for Pert's work and the discoveries it sparked were largely awarded to the very mentor that initially discouraged her from doing that work in the first place. But hey, she didn't have to pay him back for the materials. That's all for now. If you're interested in more deep dives like this, or some of the topics covered in this video, then please check out my channel and subscribe for future content. Thanks so much for watching. I'm gonna go finish this.